neutrophils, right? So the neutrophils are neutralizing this pathogenic bacteria, releasing antigens into the environment. Then dendritic cells or macrophages or B cells, they can pick up these antigens, but typically dendritic cells, they're sending out all these branches to pick up these antigens and process them. So they're sampling the surroundings, seeing what's in the environment. So remember, dendritic cells in this case are moving to the lymph nodes, and they are just going to display what they found in the environment, right? So they're going to use their MHC class II receptors, and that's what makes them a professional antigen-presenting cells. Those APCs have exclusive rights to use those MHC class II. And what do they use using those MHC class II receptors? Well, they're able to teach other cells, so namely like naive T cells and B cells. So again, remember, naive T cells and B cells are waiting in these lymph nodes or also in the environment as well, in your circulatory system, but actually in the lymph nodes. So what's happening here is that these naive T cells from the thymus, they're going to the professional APC is going to screen these T cells and see if any of them recognize this new antigens or the antigens they found. So if there is a match, what happens? Well, this naive T cell gets very excited. It's a match. So they're able to, let's see, yep, so now they're like very happy there's a match. So say what happens when they get that match? Well, these T cells become activated, and remember, they can become T helper cells or cytotoxic T cells. So what we have here is an activated T cell because why? Well, it recognizes this T cell receptor over here is recognizing the antigen presented by the dendritic cell on its MHC class II receptor. So now this naive T cell can graduate from being naive. Now it has recognized its antigen. Now this naive T cell can finally become active. So it can become, again, cytotoxic T cell or T helper cells. Yes, there are more subtypes of T helper cells, but for this class, just T helper cells at this point. All right, so here, just for simplicity's sake, I'm showing a cytotoxic T cell. So what's happening here? Well, the thing about when these cells get activated, they undergo some process called clonal expansion. So basically, these cells are dividing and making multiple copies of themselves. So it's not just like one naive T cell just becomes one cytotoxic or one T helper cell. They actually make many, many copies of themselves. And again, I can't show all the copies to scale, but what's happening here? This is clonal expansion. Notice that they all recognize the same antigen, and now they're off and to do their thing in the rest of the body. So these are what you call effector T cells. So these are the ones that have an effect. These are active T cells. But when you have activation of T cell receptors, that's not the only thing that happens. You just don't create effector T cell. A very small fraction of these cells that divide become something we call memory T cells. So memory T cells, what's the difference between effector and memory T cells? Well, they recognize the same antigen. So again, the same original antigen, all of them can recognize that same antigen. But what's the difference? Well, the effector T cells, these are the ones that go off to battle. And these are the ones that go patrol the rest of your body and then try to find the antigen, the pathogen, or cells infected by that pathogen, and kill those cells or sometimes kill the pathogen. I think there's increasing evidence that cytotoxic T cells can also target bacteria, even though we know that they do kill virus-infected cells or infected cells as well. So effector T cells, these are the ones that are active, but once they're off to battle and the infection subsides, then they also undergo apoptosis, so they die eventually. Because why? Again, when the pathogen is not there, do you really need this immune response? It takes energy, it takes time to maintain cells. So if you don't need to mount an immune response, it makes sense that your body is going to pull back and kill these cells. Now, memory T cells, notice that they didn't go off to battle, but again, they recognize the same antigen, right? So memory T cells, rather than being active and out into the workforce, they're kind of like people who delay graduation or take a gap year. They're not ready to enter the workforce, but they're still learned. They still recognize that antigen. So they still have the potential to become effector T cells. They're just taking a pause. So these memory T cells, these are the ones that hang around after the effector T cells that originally combated the initial infection. These are the ones that hang around, so they survive. So does that mean they stay asleep forever? Well, no, they hang around, and again, some of them last for maybe weeks, some last for years. Again, this is why different vaccines have different schedules. Sometimes memory T cells only last for like a few years, some last decades, some last a lifetime. So memory T cells, they do have various. I mean, for this class, should you really memorize like how much like what vaccines have what lifespan? Not for this class, but when you take advanced classes and when you get to your more professional degrees, yeah, you probably will need to know these schedules. But memory T cells, so back to that. Do they stay asleep forever? Well, remember, they still have those T cell receptors, right? They recognize that original antigen. So say the original infection subsides and it's cleared from your body, but maybe a few years or a few months later, you encounter that same antigen, your body encounters that same antigen. Say it's fighting off that same infection. Well, remember, it still has receptors for that original antigen, right? So this, when it encounters that antigen for the second time, this breaks up the memory T cell. So the memory T cell, again, is recognizing that same antigen, and then what it can do is generate more memory T cells, but also undergo clonal expansion and create more effector T cells. But the great thing, why is this faster? This memory T cell was already living. You don't have to undergo multiple screening. Remember how the dendritic cell was trying to go through all these different T cells hoping to find a match? When you have memory T cells, you don't have to worry about finding a match again. You already have that match. As long as you get that same antigen binding to the T cell receptor, this can activate more effector T cells. And this not only applies to cytotoxic T cells, but also T helper cells and also B cells as well. So they can also form memory cells and also have like this clonal expansion. Oh, sorry for scaring you. All right, so again, not all T and B cells become active cytotoxic helper T cells or plasma cells. So again, you do have a small fraction that becomes a reserve. So again, why? Because when, in case in the future you encounter that same infection and antigen, then that then that allows you to mount a faster response the second time. So memory T and memory B cells, these are immune cell records of antigens, and this is why like that cells that work anime, it really does. It says like memory T and B cells. It kind of puts them in the background. I think that puts them in the background too little, and it's like, hey, these memory T and B cells, they should actually become duplicate and form this clonal expansion. But enough about that. So again, why? Because you don't have to do that screening of all these T cells and B cell receptors again. Because you already have a T cell receptor or B cell receptor that does allow to that recognizes that antigen you were previously exposed to. This allows for a faster response. So again, this is a reserve of T and B cells, just like our National Guard, just like our military reserves. These are reserves that your body can deploy if that threat, that antigen or pathogen comes back.